session. Thank you.
Welcome to the RB Memorial Conference on Translational Research in Medicine, TRIM 2022. Good morning, everyone. I'm sure you're excited for the Track-2 drug delivery. As you know, that targeted treatment with maximum output and minimum side effects is now achievable with this field called drug delivery. Using nanoscale components to ensure the site-specific and controlled delivery of drugs, genes, and molecules to ensure safety, as well as efforts for a perfect treatment. The exciting field of drug delivery has made so much significant contributions. And for this track, we are going to have three eminent speakers: Professor Justin Haynes, Dr. Muniya Ganguli, and Professor Tejal Desai. Let me thank all these speakers for accepting our invitation and to grace this occasion. Let's start the first session with Professor Justin Haynes.
Ladies and gentlemen, it's an absolute pleasure to welcome Professor Justin Haynes from the Johns Hopkins University School of Medicine. Professor Haynes is the Lewis J. Ott Professor of Ophthalmology at the Wilmer Eye Institute at Johns Hopkins. He pursued his PhD in chemical engineering from Massachusetts Institute of Technology and has completed postdoctoral training in oncology and neurosurgery at Johns prior to beginning his faculty position in the year 1998. He is a leader of research at the interface of nanotechnology and medicine and is the director of the Center for Nanomedicine. He is also the director of therapeutics for the Institute of Nanobiotechnology, which includes more than 170 faculty members and spans several divisions of the university. His group aims at characterizing the barrier properties of the mucus layer coating the eyes, designing new nanomedicines that bypass the mucous barrier of the eyes, producing safe synthetic nanoparticles that efficiently deliver therapeutic genes to various cell types within the eye, and testing these various new systems in animal models of eye disease with collaborations from the Wilmer Eye Institute and elsewhere. He is an inventor of more than 20 patents relating to controlled, sustained drug delivery methods. Products based on his drug inventions have been tested in various clinical trials through and including phase 3. With these words, we welcome Professor Justin Haynes from the Johns Hopkins University School of Medicine.
Hello, my name is Justin Haynes from the Johns Hopkins University in the United States. I'm grateful for the opportunity to give this talk in the honor of the late Professor Banerjee, a leader in the field of nanomedicine and a beloved clinician scientist who worked at IIT Bombay prior to her recent death from post-COVID-19 complications. Professor Banerjee was taken from all of us much too early, but her legacy lives on, and I'm very grateful to be part of this conference in her honor. I'm going to tell you today about some of the work that we've been doing at Johns Hopkins on drug and gene delivery to difficult to reach locations in the body. In particular, I'm going to highlight a couple examples of translation of technologies from the concept to and including some FDA-approved products. Before I get started, I want to tell you about a center I started about 12 years ago called the Center for Nanomedicine, which is at the Johns Hopkins University School of Medicine. We're really bringing together engineers, scientists, and clinician scientists under one roof with the goal of training the next generation of leaders in convergent science that can advance both science, basic science, but also potentially create new products that can help human health. As part of this, we've been very successful in launching companies from our center. And for example, I believe it's now more than 10 companies have been launched in about the last 10 years. And more than $700 million have been invested in these companies to create products that can help people. We have multiple FDA-approved products based on the technologies from our center, and we have clinical trials ongoing or planned for greater than 20 different diseases across a wide array of conditions that including ocular diseases, diseases that affect the central nervous system, fibrosis, diabetes, HIV, COVID-19, and more. I have a couple financial interests to disclose related to this talk, and that is I am a founder and I own stock in companies shown here, Kala Pharmaceuticals and Graybug Vision. And my conflict of interest is managed by the Johns Hopkins University. So I wanted to start with just one slide on what was my introduction to translational research in this field. And that came when I was a graduate student at MIT working with Bob Langer and in collaboration with David Edwards. And we were working on a novel technology called large porous particles. And these were particles that you could inhale that, because of their large size, had a low surface area to volume ratio. And because of their porosity, however, they were still very light. Those things together allowed these particles to break up from one another from a dry powder inhaler and very efficiently be inhaled into all the way into the alveolar sacs, where they could deliver drugs from the lung airways into the blood. For example, because of this unique architecture and size and porosity, we were getting things routinely above 60% respirable into the lungs, whereas the standard in industry at that time was about 15%. Also, because they were so large, they would not be removed so rapidly from the alveolar sacs, so we could get long-term release of drugs from these particles. For example, we showed in this paper in Science that we could deliver insulin into the blood of rats for 96 hours continuously from a single inhalation, whereas the longest prior to that was 6 hours using liposomes to deliver the insulin. We also looked at things like coatings with lung surfactants and showed that they could have an impact on the performance. Long story short, this was a really amazing story, and Bob and David started a company called Advanced Inhalation Research in 1998. And then after just a few $1 million was invested into that company, they sold it to Alkermes for $115 million in 1999. This technology, the LPP technology, was also used in the founding of Civitas Therapeutics, which was then later bought by Acorda for $525 million. And it was Civitas that Acorda, I guess, that developed the first product to come out of this technology. In December of 2018, FDA approved by to treat Parkinson's disease was inhaled L-DOPA in the large porous particle format. Why is this so important to me? On the concept of cool science that challenged long-standing dogmas in the field. And it convinced me of the importance of working on things that could help ensure that new technologies that we develop in the labs could be translated eventually into products that could help people. So in my lab, in the earlier days, we became very interested in looking at ways to improve drug delivery to mucosal surfaces of the body. So at mucosal surfaces of the body, there's mucus in all those surfaces except for those with skin. So what I'm talking about is the surface of the eye, the respiratory tract, including the nose, sinuses, trachea, lung airways, the gastrointestinal tract from your mouth all the way to the other side, the female reproductive tract and inner ear, etc. So this mucus is there. It forms a very strong barrier that can protect the body against constant assault by infectious and toxic agents. But it also traps and removes other particulate matter. For example, if you try to develop a nanoparticle system to deliver to the mucus, to mucosal tissues, it gets trapped in the same mucus and can be rapidly removed. To give you an idea, mucus is replenished rapidly in these surfaces. For example, on the surface of the eye, we blink about every 6 seconds. That squeegees off a layer of mucus and everything trapped in the mucus, and it drains into your lacrimal duct in the inside of your eye and goes into your nose and you swallow it. Respiratory tract, the mucus is cleared on the order of a few hours, same with the GI tract and the female reproductive tract. So things do not last long if they are stuck in mucus in your body. So a little bit more about this mucus barrier. I mentioned that it coats entry points to your body. It's highly viscoelastic. And let me just focus on the viscous part of this because most people understand that the best. Water has a viscosity of about 1 centipoise or 10 to the minus 2 poise. In contrast, something like glycerol, which is very viscous, has a viscosity about 1,000 times that of water. Well, normal human mucus at low shear rates has a viscosity about twice that of glycerol, or about 2,000 times higher than that of water. If you have a disease, for example, people with cystic fibrosis, the viscosity of their mucus can be 100,000 or even 1,000,000-fold or more higher than that of water. Needless to say, these viscosities are much too high to allow nanoparticles to move through the solid-like portions of mucus. However, it is appreciated that mucus is a gel, and so it is not all solid and not all parts of it are equally viscous. The gel has pores in it. And for example, in this nice work by Saunders et al., they're showing pores on the order of 100 to a couple hundred nanometers or so in human mucus. And so the question we had is, could you make something small enough that could fit through these pores, and therefore could it penetrate deep into the mucus layers and avoid being cleared so rapidly? However, you also have to realize that mucus can trap things not just by sterics, but also by sticking or adhesion. So you'd have to make those particles non-adhesive for it to work. At least that's what we thought. What was interesting was the major dogma in the field was that you actually wanted to stick to mucus in order to get things to last longer. They call that mucoadhesive. But we thought that maybe you have to get through the secreted mucus first before you could stick to the underlying epithelium. And so maybe that dogma might not always be correct. So long story short, skipping through quite a bit of work, Sam Lai in my lab in 2007 published this paper where we finally showed that you could take conventional nanoparticles, which don't have any kind of coatings, so they just stick very readily to mucus. And this is a video of the particles not moving in mucus. So little bright things with the nanoparticles, and you can see that they're just stuck. On the other hand, if he had very dense coatings, specifically with low molecular weight polyethylene glycol, then the particle surfaces were neutrally charged and very hydrophilic. In that case, they did not stick to mucus. And in fact, we've shown that we can do this. We can make particles that penetrate all different types of mucus, but from both animals and even human mucus, from the lungs, eyes, sinuses, cervical vaginal tract, and the GI tract. Key to that is not only the size, they have to be nano-sized, but also that high peg density. And I'll just show this really quickly. Even with quite a good amount of peg on the particles, they would move orders of magnitude slower. This is looking at the percentage of particles moving with a speed called the effective diffusion coefficient. You can see that the particles that have a low amount of peg, shown in the clear bars, are orders of magnitude slower than most of the particles that have the very dense peg. Okay, and so we showed that this is actually the peg density here is well into the brush regime in order to get the particles to move. But once you get enough peg, it's like a light switch. They go from being stuck to going through mucus very rapidly. So then with these kind of particles in mind, I wanted to ask this question: which particles adhere to mucus in this picture? I'm showing a GI tract. First, I'm showing a cross-section of a human GI tract. So you can see that when it's not full with food, its pressure squishes it down. And so it's highly folded. And the white part is the lumen. And then the rest is like the tissue. So here's all the lumen areas. And then in a rat, you can see, or this is a mouse, or oh, it's a mouse. Okay, in a mouse, you can see it's kind of a similar. The blue is the staining of cells. So you can see the epithelium along here. In both cases. So one of these cases, we administered green particles, and the other we administered red. One is sticky to mucus and one is not. And the question is, which one is which? The dogma would say that the ones that stick should give you better drug delivery. But in looking at this picture, it looks like the ones in red here penetrate much better, get a very even distribution over the entire epithelium. And in fact, we found out the ones that are red last much longer. Well, the ones in red are not mucoadhesive. They're actually the ones that are not mucoadhesive. They are non-mucoadhesive. So the ones that do not stick to mucus are the ones that spread well. And in fact, my lab has gone on and we've published many papers since that early 2007 paper and shown that we can make mucus penetrating particles that behave this way in all kinds of areas, like the GI tract, the lung airways, the female reproductive tract, whereas particles without the dense PEG coatings, what we call conventionals, get stuck and more rapidly removed from each of these cases. They don't get as near to the epithelium, and they get more rapidly removed. And we've also shown in many of these papers greatly enhanced drug delivery from a PK point of view, as well as an efficacy point of view, and also greatly enhanced gene delivery in many different organs. So we're very excited about this technology because it can be used to treat, a wide range of different diseases. Here's just a few that I'm listing in some of the potential impact that it could have in these various areas. This technology was licensed to a company that I started called Kala Pharmaceuticals. And this mucus penetrating technology, mucus penetrating particle technology, or MPP, is now called Amplify by Kala, to give it a cooler name. And that's the technology that's now in Kala's ocular drug delivery products, Inventus and Isuvis. So I'm going to tell you just a little bit about those as well. So about the time I started that company, which was December 2009, I started working at the Wilmer Eye Institute at Johns Hopkins. And we were interested in trying to see if these mucus penetrating particles might also help delivery to the eye. So first question was, can we get particles that penetrate eye mucus? So we got some human eye mucus. And first of all, if the particles were uncoated, so typical nanoparticles, they would get stuck. And actually, in this case, they got balled up into large balls that became large and bright. But you can see that they're stuck and don't move. So those could be easily blinked away. Whereas the particles that were coated do not get balled up, and they go in and out of the field of view, meaning they're moving several microns on the order of seconds. So could very easily penetrate human eye mucus, in other words. And then the question was, well, we blink so rapidly, could this fast movement be enough to get them deep into the mucus layers so that they won't be shed off so rapidly when you blink? And so this is where Kala Pharmaceuticals, the company that I had started doing some work on it, and especially Hongming Chen and her group there. And what they found was, again, if you had conventional particles here on the left, so just nanoparticles that don't have the coatings of drug, and you looked at 2 hours, 4 hours, and 6 hours, that most of the particles were gone within 2 hours, and almost all of them were gone by 6 hours. On the other hand, if you took the same exact drug particles and coated it with PEG, then you had many more at 2, 4, and even quite a few at 6 hours from a single drop. They did pharmacokinetics, and this particular drug is called loteprednol etabinate, and it's a soft steroid that was found in the product Lotemax. So they compared their version of this, a nano size version of lotipretinol etabinate with the non-mucoadhesive coatings. And they compared that to the Lotemax, which was the leading drug that was approved at that time. And what you see here, they looked at various eye structures, but I'm just showing the cornea for simplicity. If they have it in the MPP version, then they got much higher levels early on than Lotemax achieves. And that was sustained over quite a long period of time, such that the improved delivery was something like 40-fold the area under the curve, I believe is what Dr. Chen had mentioned to me. So it greatly enhanced pharmacokinetics of this drug. This led eventually to the approval of this drug in a couple different formats. One is called Inventus. It's their 1% lotipretinol etabinate suspension, nano suspension. It was approved in August 2018 to treat ocular pain and inflammation. It's the first and only post-surgery ocular steroid that can be given just twice per day, BID dosing. Previously, the steroids, lotipretinol, I believe, was 4 times per day. So this was a big advantage to go to a system that only needed to be delivered. Also, the sustained release that was achieved led to low rates of adverse events. In particular, one worries with steroids about potential for increasing ocular pressure, which can lead to glaucoma. But what they found was that intraocular pressure was similar in patients who took Inventus to just patients who got a vehicle. So similar to placebo, which is a huge win, I think. This drug is now indicated for all ocular surgical procedures, of which there was about 8 million of those in 2017. They've also shown that the same drug, but at a lower concentration, can be used and as a potential first-line therapy to treat dry eye disease, in particular dry eye disease flares. Dry eye disease is actually a terrible condition that affects about 33 million people in the United States alone. And about 90% of these patients experience what are called as flares. In other words, they don't have really bad symptoms all the time. Just once in a while, all of a sudden their dry eye gets really bad and they need a medication. So Isuvis can be used in those cases. It was approved by the US FDA in October 2020. So we're excited about these products and Kala's developing more, but we're also still developing mucus penetrating particles for a variety of other conditions. In particular, these things I'm doing largely with my collaborators and former students who are now professors at Johns Hopkins as well, Professor Laura Ensign and Professor Jung-Soo Suk. And we're interested in things like the prevention of sexually transmitted infections. I just give an example paper here, Laura's paper when she was a graduate student, which was the seminal efficacy paper with MPP technology for showing that it could be very effective in preventing herpes simplex virus infections. Prevention of preterm birth and the neurological deficits associated with that is some work that I'm involved in, but really led by Professor Ensign these days. So there's some work there, and many other things: lung inflammation, lung cancer, inflammatory bowel disease, and more. So there's a huge number of potential applications of this technology. A second technology I wanted to tell you about is this long-acting non-inflammatory technology that has been licensed to a company called Graybug Vision. While Kala is making drugs that can be delivered topically to the eye, those types of drugs typically treat the front of the eye conditions. But if you have a disease that affects the back of the eye, like the retina, typically people take injections for that. And for example, in one of the most common blinding diseases, age-related macular degeneration, people at the time we started developing this technology, people were taking shots about once every month into their eye of an anti-VEGF agent to treat AMD. And so we wanted to develop something that can last a lot longer than that. And it was, when I moved down to Wilmer Eye Institute to start the center, I was puzzled why people weren't using polymer microparticles to make these drugs last a lot longer. Well, what I found out was that people said that, all of polymer particles cause inflammation when they're injected. You don't always, but by the time you get up to humans, the inflammation occurs. And so we became interested in this, and we wanted to develop something that could be non-inflammatory. We can inject. If you're injecting particles, those particles also need to aggregate into something that would not float all over in the eye, which could disrupt vision, and would need to release drug over a long period of time. So that was the idea, but that first major hurdle was, could you make them non-inflammatory? And again, a long story short here, but we found that if we had very dense PEG coatings again, that we could prevent the adhesion of the drug particles to the intraocular gel in the vitreous. And preventing that adhesion appears key to preventing particles from causing inflammation inside the eye. So I'm just showing here a patent that we had, and you see the first name listed here is actually McDonald. That's Peter McDonald, the head of the Wilmer Eye Institute at that time and still. And he was a collaborator with us on this and testing this in animals. And so we showed that basically if you have particles that were non-coated with PEG, or even particles that have pretty good coating but not very, very densely coated, then you had quite a bit of inflammation after injection into various parts of a rabbit eye, including intracameral injection, which is a very sensitive way of testing this. On the other hand, if you had the particles that were surface treated, including a very dense coating with PEG, and injected them, then the inflammation profile was just like injecting saline. So they were non-inflammatory and at least in animals at that time. And this technology was then licensed to the startup I told you about called Graybug. And so Graybug is a company that I founded along with Peter McDonald and Peter Campachero and also Christy Weiskill. And we had to select then a drug for this. And our lead, actually our second lead drug, that's why it's GB102, but what is now Graybug's lead drug, GB102, uses sunitinib. And why did we select sunitinib? Well, sunitinib is a pan-VEGF receptor inhibitor. Okay, so it blocks VEGF receptor associated with angiogenesis, vascular permeability, cellular proliferation, and fibrosis. So in addition to being anti-VEGF, which could be very good in things like AMD, diabetic retinopathy, and macular edema, sunitinib is also known to be strongly neuroprotective. Some work, beautiful work done by Don Zach from the Wilmer Eye Institute, elucidated that it's a DLK pathway inhibitor, and that blocking that promotes retinal ganglion cell survival. And so it could also be useful in things like glaucoma and various retinopathies. So for this reasons, and more actually, I mean, another one is that sunitinib had been used for a long time as a cancer drug orally. And they found when it's given at these high doses orally, quite a bit of it would end up in the eye. And when it did end up in the eye, it did not cause any toxicities or problems in the eye. So it was another reason we liked it. Jay Fu from my lab I've worked with her for many, many years, and she's one of the best formulation chemists I've ever met. She made delivery systems for sunitinib that using PLGA systems with dense coatings of PEG that could release sunitinib in a very steady fashion for as short or almost as long as you'd like. So from 30 days to well over a year. And I'm just showing this, but there's a lot of preclinical, we published a lot of that in Nature Communications in 2020 with Peter Campachero, myself, Ming Yang, Jay, and others, many others. So you can look at that if you'd like. But I'd like to show you a little bit of what Graybug has seen in the clinic. In the clinic, this is now looking at photos of actually the drug after it's been administered into people's eyes by injection. And this is a 1 mg dose of GB102. And the particles show up in this kind of light yellowish, orangish way. They're out of the field of view normally, but we could take pictures of that area. And so this is a baseline. You can see the particles aggregated in out of the view down here, 2 weeks, 1 month, 2 months. They start to finally degrade 5 months, 6 months. You can still see there's some color there and almost completely resorbed though. What was remarkable is that once we started injecting this in patients, and now it's been injected in, I believe, over 100 patients, there's been no inflammation that I'm aware of that has been seen in any patient. So we were able to solve that problem so far. And it looks like this is lasting for a long time. And in fact, some of the early data that was coming out was showing that it was very durable. This anti-VEGF effect of GB102 is very durable and could last through at least 8 months from a single intravitreal injection. And this is just looking at a couple of ways that clinicians look at that, and including the best corrected visual acuity and the central subfield thickness being very stable over time in these patients. This is one patient. What did it look like for more patients? This is from Graybug's phase 1/2A trial in age-related macular degeneration, and it's going to be looking at how long rescue-free survival, meaning how long after a patient getting one injection of GB102, how long did they, how long was it until they needed another injection of medication? And so what we're finding here is, this was with a variety of different doses, and this is the overall kind of probability of not needing rescue, and looking at 3 months, 6 months, and even 8 months. But what was key was that the best dose, the 1 mg dose, lasted greater than 6 months in a great majority of the patients. About, in this phase 1/2A study, in about 88% of the patients. It lasted for 6 months, which was a lot longer than anything that exists. Right now with Eylea, the average time for retreatment is about 6 to 7 weeks. It's on label for every 2 months, but in practice, the average time is about 6 to 7 weeks. So Graybug was founded out of my lab, I mentioned, and along with some others, but some people have asked us, where'd you get this weird name? And so this is a picture taken about the time that Graybug was founded. And at that time, you can see I had very young kids. In the bottom left here, Grayson, who we called Gray, and on the bottom right here, Riley, who we called Bug. And so when I started this company, I wanted to name it after them so that they wouldn't be, too upset if I had to be working long hours while I'm working on this company, as well as my normal day job at Johns Hopkins. And this is just showing that Graybug went public in September of 2020. The last thing I want to tell you about is changing gears from the eye and talk about some drug delivery in the brain, another difficult to reach tissue. And so back in 2012, this is all tied together because Elizabeth Nance, Graham Woodworth, and others in the lab started testing whether or not particles that had very dense PEG coatings that were non-adhesive, whether or not they could spread out better once they were inside of the brain. And that is a key limitation to current therapies. When they're given in the brain, they just stay wherever they're administered and they don't spread too far. So we wondered if we could get things that would penetrate that brain tissue better. And so I'm showing obviously some results of that in 2012 of our early systems, where the green particles are the ones that are coated and the red ones are the ones that are not coated. And you can already see that I'm giving away the results. But what was interesting was the dogma was that particles, to be able to spread, they would need to be smaller than about 40 to 60 nanometers, because the extracellular matrix mesh size in the brain was about that. But what we found was if we co-injected unpegylated particles, which are UPN or red, and densely pegylated particles, which we call brain penetrating nanoparticles or BPN, if we co-injected those straight into the brains of mice, for example, and then looked at them, if the size was about 40 nanometers, you can see that the BPNs could start to diffuse away from the injection site, but The uncoated particles just stay right at along the needle track, right here in the yellow. But even 100 nanometer particles, the brain penetrating nanoparticles could spread away. And we had a intracranial window on this, and we could do some confocal microscopy and look at a region away from the needle track. And you see that the particles that are densely pegylated and even 100 nanometers are still moving rapidly by diffusion. And in fact, as we find out later, they can spread quite some distance in the brain. If you got up to 200 nanometer particles, it didn't matter if they were coated or not. They were now too large to make it through the space between the cells and so forth. And so they would all be stuck and along the needle track. So from there, we went on and made particles that had different types of drugs. And I'm looking at some work that was done by Clark Zhang and others in the lab, where we used nanoparticles that had cisplatin, what we're calling CDDP, but cisplatin here. And it either administered these uncoated particles or the densely coated particles that penetrate into the brain, either just as a bolus or by convection enhanced delivery, where it's slowly infused over time. And what you can see is that if you have the coatings and you administer by convection enhanced delivery, you can get a greatly enhanced volume of distribution. The particles can spread widely throughout the brain from that infusion, as compared to if the particles were standard particles without the coatings. So the brain penetrating nanoparticles really spread well. Clark and others in that work, notably a collaboration with my former postdoc mentor, Henry Brem, and also my former co-worker during my postdoc, Professor Betty Tyler. Clark showed that these brain-penetrating cisplatin nanoparticles could cure animals that had established F98 brain tumors. So these are rats. F98 is a tumor model. And what we found was that if you don't treat the animals at all, they all die by about 40 days or so of their tumors. If you gave the unpegylated particles, it was better than doing nothing, and we actually got some survivors. But if you gave the pegylated particles that could spread better, we had 80% survival. Notably, if we infused locally the free drug cisplatin, you got some long-term survivors, but you actually killed most of the animals early due to toxicity. So this would not be an acceptable therapy. We've also worked on doing gene carriers, and Jung-Soo Sak, when he was a graduate student in my lab, actually figured out how to make particles that could be very small and carry a large gene and still have very dense peg coatings. And this was done through a method of blending of different types of polymers. You can see some of that work even in Masteraka's probably cites it and so forth. But I don't have time to go into that. But this is just showing now, we started to apply these into the brain. And again, if it's a gene carrier and it's small and has dense peg coatings, that's shown in red. It's spreading quite well and having a high volume of distribution in the brain. If the particle has PEG, but not a lot of PEG, we call those conventionally pegylated nanoparticles. They don't spread quite as well. They're showing up here in yellow. Okay, and here's looking at the volume of distribution. So in fact, when you infuse these particles into, for example, a rat brain into the striatum, one infusion can give you nanoparticles going almost throughout the entire rat striatum. And this is now looking not just at where the nanoparticles are, but where you're actually seeing a gene product being made. So we put a gene inside of the DNA that would produce a fluorescent molecule so that you could see that under fluorescence. These are the 2D images. You get many of those, you can make 3D images of it. You can see it's throughout the brain. So this could be, local infusion can be done in people, for example, with brain tumors. They have surgery to remove their tumor, you can leave a catheter behind and you can infuse drugs through that. But for a lot of other diseases, it would be better if you could do non-invasive delivery into the brain. So I've been collaborating with Rich Price at the University of Virginia for about 20 years or so. And Rich is one of the absolute leaders in the world in using focused ultrasound combined with microbubbles to open up transiently, to open up tight junctions and allow drugs and nanoparticles to get into various tissues. And in fact, Rich showed, and we collaborated with him by giving him some of these nanoparticles, these BPN, brain-penetrating nanoparticles, showed in Journal of Controlled Release in 2015 that you can use this focused ultrasound to even get nanoparticles to go from the blood into the brain. And the way that works is they're co-infusing these albumin microbubbles, and they're just large albumin bubbles with air in the middle. And when you select an area of the body to give focus ultrasound with sub-millimeter precision, you can cause a pressure wave. And that pressure wave is doing locally is just causing these bubbles then to oscillate, kind of collapse and then expand, collapse and expand. And if you do that under very controlled conditions, then you can get this transient opening of various tissues, including the blood-brain barrier. And by the way, I should say this is done safely. It's done safely in hundreds of humans now as well. But in the early days, we were working on this or Rich was using our particles, and he was doing the lion's share of this focused ultrasound work, of course. And we gave him some of our gene carriers, and he put them into the blood, and then used focused ultrasound to open up a spot in the rat brain, and then looked for whether or not he was getting transfection. So again, this is, we're delivering a gene that would produce a fluorescent product. And this is the first study, actually. And so it was pretty remarkable. You see a dose response here. When we give 50 micrograms, you see a little bit of a gene transfection with the brain, 100 micrograms more, 200 micrograms more, 300 micrograms, 350 micrograms even more. When you take out the brains, you can see it even more clearly here on the bottom. What was also remarkable is you see very little transfection anywhere else, including the liver or other tissues. Very little or actually not perceptible. So we've been working on this in a variety of different drug therapies and gene therapies. One thing I'll just highlight is some work that we published a few years ago. One of Rich's students, who's now a PhD himself, Dr. Mead, did this work where we delivered a gene for GDNF, which is a neurotrophic growth factor, into the brains of animals that in a Parkinson's disease model, where one half of their brain, the dopaminergic neurons have been poisoned. And so they're missing those. And what we found was that with a single dose of the nanoparticles delivered into the blood, we can get them into the brain and get very high levels of GDNF expression in that side of the brain. That high levels of GDNF could then cause the dopaminergic neurons to be restored at those local sites, both in the striatum and also in the substantia nigra pars compacta. That would lead to motor neuron density being restored, at least up to about 70 or so % at week 12 of the normal levels of motor neuron density. Moreover, that these motor neurons were functional, so they were actually producing dopamine, and so the dopamine levels were restored, again, up to, this is 6 weeks, 12 weeks, it's getting even better, almost back to normal levels shown in the gray here, or about 70 or 80% of normal levels of dopamine. And that this led to efficacy in a couple of different Parkinson's disease models. Apparently, I don't have much time to go into, unfortunately, but one is called a contralateral rotation test, in which animals with one side of the brain poisoned, they will spin around in their cage a certain number of times per minute. But if they were treated with the treatment, actually started doing that less and less over time and got down to a much lower number, whereas all the control groups got worse, progressively worse. And the same thing, this is a contralateral touch fraction. A jar is put over the animals, and when they're in that jar, they try to feel their way out. If it's a normal animal, they'll touch the jar equally as often with their left hand as their right hand. But if it has this model of Parkinson's, then they have a bias towards one part or the other, and that's shown here. Like it's touching one side, 3 times for every 1 time it touches with the other paw. But in the animals that were treated, even though they started out just as bad as the others, they went back to more like normal. I looked over and the number of people involved in just the work that I've talked about here was well over 100. And so there's no way I could acknowledge everybody. And I hope I have not missed too many people here. But I wanted to list some key collaborators over the years, both in academia, at Hopkins and other places, in industry, of course, many people in their teams, Rich and many people from his teams, many students in my lab, including these and many, many more have been involved, and also thank the NIH for funding. Thank you so much for your time and attention.
This was quite an interesting talk by Professor Justin. Let's thank him. And however, due to some unavoidable circumstances, he is not able to join today. I request participants, if they have any question, they can type the question in the chat box with their name and email ID, and we can send your questions to Professor Justin and revert back to you with answers. Moving ahead, let's welcome the next speaker.
Thanks, Ayushi. Should I load my slides now?
Yes, ma'am. You can share your screen and we can start the presentation. Sure.
Sure. Is it visible?
Yes, ma'am, it's visible. You can make the presentation full screen.
Yeah, is it full screen now?
Not yet.
Okay. I'll try once more. Now?
No, ma'am.
Full screen करूँगी. Full screen के थोड़ी करूँ. दोबारा से कीजिए. Share screen. Entire screen. Share. 50 select कीजिए. हाँ, इसको full screen कीजिए.
Okay. Is it okay now?
Yes, ma'am, it's okay.
It's okay.
We can see your slides.
Okay. All right. All right. Yeah. Good morning. It's an honor and privilege to be speaking at this conference today, where I'll be presenting my lab's work on peptide-based nanocomplexes for gene delivery to skin and lungs. This is my first talk actually at IIT Bombay, although it's a virtual presentation. The last time I had an opportunity to speak at IIT Bombay was an invitation from Professor Banerjee, and I feel really bad that I could not make it because of some personal commitments, and I so wish I had done that. And I have some fond memories of Rinti. She was an external examiner for one of my PhD students way back in 2013. She was a very critical evaluator of the thesis, but very kind as well, pointed out all the mistakes to the student, very gently. And we have kept in touch through the years off and on. And in 2019, when, the connected academic that she was, the special issue of ACS Biomaterial Science and Engineering that she edited, she had requested for an article which we published in that volume, and we interacted quite a bit at that time as well. It's really sad that we will not be able to continue our interactions anymore, but in a way, this is my tribute to her. This talk is my tribute to her. So I come from this institute, the CSIR Institute of Genomics and Integrative Biology. It started as a very small center for biochemicals in the 60s, and in 2002 it was rechristened as the Institute of Genomics and Integrative Biology, and in the 20 years it has become one of the leading genomics institutes of the country, and also took part very actively in the COVID-related activities, be it diagnostics or surveillance, genome sequencing, serosurveys, and so on. In this institute, my lab is in the Chemical and Systems Biology unit. And what we do in the lab is primarily we design materials for nucleic acid delivery. So be it for cellular transfections or for delivery to multiple tissues with possible gene therapeutic applications, and different formats of nucleic acids, plasmids, small RNAs, and currently also going into mRNAs and RNP complexes in the context of CRISPR. And so basically, we design these carriers, form nanocomplexes with different types of nucleic acids, and try to see, what properties of the carrier are important. Our lab also works on surface functionalization of different inorganic and organic nanoparticles for a wide range of applications, and also skin being an organ that we are very much interested in. We also look at delivery of different large macromolecules to skin. I will not be touching upon these aspects of the lab work, but only on the nucleic acid delivery part. Now, why nucleic acid delivery? Why gene delivery? And this is something that you all know, but still I will stress upon it a little bit. So for any gene therapeutic application, be it for gene augmentation or gene silencing, gene editing, you need carriers, and that is the key challenge in making a gene therapy succeed. One of the key challenges. And over the years, as you will see here, after some early setbacks in the 90s and early 2000s, it has come a long way. We now have about 24, 25 products in the market, gene therapy products in the market. But getting a good carrier still remains a challenge. And viral carriers have been and still continue to be the major, the major type of carriers. Lipids are also quite extensively used, especially in the context of vaccine delivery now. And but still they come with their own limitations and challenges, and therefore there's always a necessity for developing better carriers. And why it is challenging? Because the carrier which takes the nucleic acid in vivo has to face a large number of barriers, and these barriers range from organ level barriers, where the nanostructures that take the nucleic acid in, the nanocomplexes or nanoparticles, they will be sequestered through in the liver. There will be protein corona formation when they travel through the blood, and also sub-organ level barriers, like even after it has moved through the blood vessels, it has to come out from them to reach the target organ, and in the process it will meet the barrier of the extracellular matrix. And after it reaches this intended cell type, it has to be effectively taken in by the cells, and inside the cell also it has to come out of the endosomes to avoid getting degraded by the lysosomes, and then it has to target the organelle where it is intended to act, the nucleus in case of DNA delivery. So the carrier that you are going to design, the size, shape, surface modification, and so on, has to take care of all these different barriers in order to make the final delivery successful. And as I mentioned that, viruses have been the most important carriers for nucleic acids, but they have their issues with, the size of DNA that they can carry, or in some cases, the random integration that might happen, immunogenic reactions. So there is a lot of stress on developing non-viral carriers for gene delivery. And there's a wide range of materials, polymer-based, lipid-based, mixture of them, nanoparticles, hybrids, exosomes, and multifunctional devices. Basically, they operate around the same kind of principle. They complex the DNA or encapsulate the DNA or other nucleic acids, and make sure that the size, charge, stability, etc. are so optimized. Sometimes it needs a coating as well, that the systemic distribution and the stability against nucleases, their biodistribution, clearance, all these parameters are optimized. And inside the cell, all the barriers that I talked about are overcome as well. So you need the carrier to be efficient. Also, it has to be non-toxic, and it has to be easy to synthesize as well as administer. So in this context, other than in the non-viral space, other than lipids and polymers, which are most well known, cell-penetrating peptides have also come a long way. What are these cell-penetrating peptides? These are short peptides, less than 30 amino acids in length in most cases. They could have either they could be fully positively charged, or they could have a mixture of positive charge and hydrophobic residues, which make them amphipathic, both primary and secondary. And these are basically derived from some proteins, the TAT being one of the most common cell-penetrating peptide that has been extensively studied as a carrier. Also, there are several synthetic peptides where the sequences are derived from the natural peptides, and they are very versatile in terms of both the mechanism by which they enter the cells and how they carry the cargo. So usually the cargo is attached, it's conjugated to the peptide, and they can enter either by energy dependent processes like endocytosis, or by other energy independent processes like pore formation, or formation of a carpet-like structure on the cell surface, or inverted micelle formation, and so on. But in the context of nucleic acid delivery, it is important to have good cellular penetration, but there are several other parameters that one needs to take into account, even to overcome just the cellular barriers. So if you look at other kinds of peptides, yes, there are different classes of peptides like DNA condensing cationic peptides or endosmolytic peptides, which can, break open the endosomes through the proton sponge effect, or there are membrane perturbing peptides, peptides with nuclear localization sequences that help in the nuclear entry. So what we wanted to do is to sort of combine all these different properties of peptides that will help overcome the barriers of cellular entry and make single peptides. So I will, so this work was primarily done over a period of 4-5 years by Anita, Rangita, and Rajpal, and Daniel, Vasundhara, and Shivangi were ably assisting these PhD students. And I will just spend a couple of slides explaining the two systems that I will be talking about in the later part of the talk. And one was a set of fully cationic peptides that we designed. So we optimized the number of positive charges, arginine being the residue that we, being the amino acid that we chose, not only because it can condense the DNA because of the positive charge, but also the guanidinium head group that it can interact with the cell membrane as well. So they will fulfill the property of both interacting with the DNA as well as or any other nucleic acid as well as membrane interaction. We also optimized a number of histidines that are necessary. Why histidines? Because histidines have this imidazolium group, and they help in the endosome buffering and pushing the cargo out of the endosomes into the cytosol. We also use cysteines at two ends of the peptide to give a bit, good condensation release balance. So the cargo, the nucleic acid, when it interacts with the peptide, it is condensed, but inside the cell, it has to release also, right? So cysteines, what it will do is that you have an SH, and it can form SS bonds when you condense the nucleic acid, and in the reducing cellular environment, the SS bonds will break and the cargo will release. So that was the idea, and that is how we designed these. And just to show some snippets of the results, these are the particles that were formed, like 50-60 nanometer size particles. And we usually, whenever we measure the transfection efficiency, we do it with the reporter gene. In this case, it is luciferase. And we see that the transfection efficiency overall is as good as commercial agents. And this is one interesting data that, if you use a proton pump inhibitor like bafilomycin, the transfection drops, which means that, the endosome buffering property of the histidines works. And if you do not allow the protons to be there, then the endosome release decreases drastically and the transfection drops. So this was one set of peptides that we designed. In the other set, that is an amphipathic peptide, what we did is we retain these arginines, histidines, and cysteine. We reduced the number of histidines a little bit. But we also added some hydrophobic residues and made it a secondary amphipathic peptide. Now, why hydrophobic residues? To improve the membrane interaction. And secondary amphipathic because if it forms an alpha helical structure, which it does in the presence of the membrane, it will have the hydrophilic, one phase will have the hydrophilic residues, the other phase will have the hydrophobic residues. So basically the concept is similar, that it will form these stable complexes with the arginines and the cysteines. The histidines will help in the endosomal release. The hydrophobic moieties will help in the cellular entry. And we kept the hydrophobicity somewhat low, because otherwise, and also made it secondary amphipathic, because the primary amphipathic with high hydrophobicity often leads to a lot of aggregation. So we avoided that, and we formed these monodispersed nanocomplexes. These are also less than 100 nanometers, and it shows high transfection efficiency in most cell lines. The other good thing is that it shows very low toxicity. So these are the two systems that we developed for cellular transfections. Now, how about applying them to in the in vivo context? So what we did is that we chose skin as an organ, and this work is primarily that of Manika Vij and Purnima and Tanuja were the project assistants who helped her with this work. And why we chose skin? One is, of course, that it has a very large surface area, so you can administration is easy. You can phenotypically monitor. The access is also easy. So for these systems that we developed, if you apply them to an organ like skin, which is exposed organ where you do not have vasculature in the epidermis, you don't encounter the barriers of the blood. So it's easy to access and easy to deliver. You can also do both localized and systemic delivery. So topically on or inside the skin. So in the different skin layers, you can deliver small drugs, peptides for a large number of diseases and conditions. But you could also do transdermal delivery across the skin and send it to the circulation. And that bypasses the gastrointestinal metabolism. So it's a preferred way of delivery of a number of drugs. But it doesn't have, so skin in the epidermis epidermis, so this is the epidermis of the skin. It does not have the vasculature, so the barriers of vasculature are not there, but it has a very complicated structure. So there are barriers associated with skin in a different way. So what are these barriers? Like, so this the epidermis of the skin is covered by what is called the stratum corneum. That's the topmost layer of the epidermis, actually. And this contains these terminally differentiated corneocytes, and they are rich in keratin, and they are embedded in a lipid matrix, and they are connected by what are called desmosomes. So all these, the presence of the corneocytes, the desmosomes, the lipid matrix, it all poses a significant barrier for anything to come inside the skin and go into the epidermis. And of course, like, when we are thinking of nucleic acid delivery, it has to then enter the skin cells in the epidermis, the keratinocytes or melanocytes, and then all the cellular barriers are present anyway. So because of this kind of a structure, small molecules which are less than 500 to 800 Daltons in size, and which have a very high partition coefficient and are lipophilic, are the ones that can go in unaided into the skin. So nucleic acids obviously being large and hydrophilic and a lot of negative charge, they cannot go inside the skin by themselves. So they need assistance. There are, of course, viral vectors as well here, but for a number of skin-related disorders, common well-known disorders like melanoma or psoriasis, rare skin disorders like EB and ichthyosis, there are, clinical trials going on, academic research, of course, and then moving on to clinical trials as well for a number of nucleic acids, DNA, siRNA, ASOs, and so on, using different types of physical as well as chemical methods, lipid-based, peptide-based, and so on. So what exactly, how do they overcome these barriers? Like when you are sending the material using these carriers, how exactly do they enter the skin? So one of the pathways is through the appendages, the sweat gland, the hair follicle, the sebaceous gland. So inorganic nanoparticles and some nanocomplexes going through this route. It's not a very preferred route because one, the follicular openings on human skin is not very high, and they can also go and deposit there and not release the drug or DNA or whatever you are sending. The other pathway is by interaction with the proteins, be it in the desmosomes or be it the keratin protein, and this is called the transcellular pathway. There are several skin penetrating peptides known that use this pathway. The third, and this is the most common pathway actually, is through fluidization or through lipid extraction. So what happens is that the lipid structure that is present is disturbed or lipids are extracted. And this is how several lipid-based carriers, and there are some skin-penetrating peptides also known, which use this kind of a pathway to enter. Now, when we looked at what kind of skin-penetrating peptides are, have been used in the literature, we could not really come up with any commonality, except that several of them have arginine residues. Some of them are hydrophobic as well, and most of them form alpha helical structure. So we thought that our peptide MGP9, that is the amphipathic peptide that I talked about, it is secondary amphipathic. It has arginines, it has hydrophobic residues, it forms an alpha helical structure in presence of the membrane. So why not try it out as a skin penetrating peptide? So we first checked for its entry into the skin cells, and we compared it with two well-known cell and skin penetrating peptides, STAT and penetratin. We found that the entry into skin cells, the blue bars indicate the entry of the MGP9 peptide. It's comparable to the other two. And we also looked at the skin penetration in the tissue. So what we found is that MGP9, this is a labeled peptide that we used here, does penetrate into the skin tissue. So does STAT, but in case of penetration, we did not find any skin penetration. And it is retained also in the skin tissue for about 20 24 hours. We checked up to 24 hours and it's retained in the skin tissue. So we were very excited about this, that, okay, the peptide that we designed does act as a skin penetrating peptide. Now, can we, does it actually carry plasmid DNA? Because in case of many of the other skin penetrating peptides, the cargoes that were sent were chemically conjugated and not always complexed into nanocomplexes. So we wanted to check that. We used lipofectamine as a positive control because that's a well-known DNA delivery agent. And interestingly, we found that MGP9 and lipofectamine both show very strong plasmid DNA delivery into skin tissue. So we had to standardize the amount, etc., how much application we have to give, and that's why I'm just shown the experimental process also here. So we had to apply this for 3 consecutive days using 4 microgram of plasmid DNA in every application. And this is in the inserts that are put in 24 well plates. And TAT, on the other hand, does not show DNA delivery, although TAT is well known as a skin penetrating peptide when you conjugate cargoes to them. So this was quite interesting for us, and we next wanted to look into the depth up to which the nanocomplexes can traverse. So, okay, we saw the expression, but how deep does the DNA delivery happen? So here, this is an immunostaining experiment where the green line that you see here is actually the keratin 14 marker for keratin 14. The secondary antibodies attached to a green fluorophore. So keratin 14 actually is predominantly present in the basal layer. Basal layer is the end of the epidermis. And the red is actually the GFP signal. So here again, the secondary antibody is TRITC. So that's why you see it in red. And we had to do it in this way because otherwise the skin autofluorescence sometimes makes it difficult to actually assess the signal. So what we see is that both for lipofectamine and for MGP9, the whole epidermis, we see expression. So this is in human skin tissue, ex vivo. And in vivo mouse system also, we have a very similar result. We used a hairless mouse, SKH1 mouse, which is unpigmented and but immunocompetent, and it's hairless, so it's easy to administer. We don't have to do any shaving, etc. Also, one very interesting about SKH1 mouse is that the follicular openings is less as compared to other mice. So it is non-follicular actually. So it mimics the human skin a little more than normal mouse. And clearly we see expression here, which means that the follicular pathway is not what is operative in this case. But we see expression with lipofectamine as well. So that's a standard agent. So why MGP9? So we wanted to find out if there is anything advantageous about MGP9 that we can think of. And what we saw is that we actually did what is called a skin integrity test. So this is basically a Franz diffusion apparatus where we put the skin in between the donor and acceptor chambers, and we administer our nanocomplex along with a dye that does not otherwise permeate through the skin. So if the nanocomplex causes some disturbance in the skin, permanent change in the skin, the dye will be seen in the receptor chamber. So if you look at this data, when we administer lipofectamine, we see that there is indeed the dye penetration, which means that lipofectamine does cause some damage to the skin, whereas in case of bare MGP9 or for the complex, there is no such evidence seen. We next wanted to also, do some preliminary check on what could be the mechanism of entry, and we did an FTIR analysis of the stratum corneum on which we applied the peptide, and we see some minor changes. At 4 hours, we actually see changes in the bands of both the skin proteins as well as the skin lipids. There is disappearance of a couple of bands pertaining to both the protein amide vibrations as well as skin lipids, which are restored at 24 hours. So it probably interacts with both the skin lipids as well as the proteins, but we have to do further checks to sort established that. In a way forward, there are 2-3 things that we are doing. One is to enhance the delivery efficiency further in the skin, also moving towards a formulation. So we used a new material, silicone oil, which has not been used in the context of nucleic acid delivery, and we found that the expression is further enhanced with silicone oil as compared to known chemical enhancers like SLS phenylpiperazine, where there are some changes in the skin structure that happens. We are also trying to modify the peptide with ligands such that we can make them enter specifically into one cell type in the skin. And we have some preliminary encouraging data on that as well. But these are all like, all with reporter gene expressing plasmids. So we do have the MGP9 working as a very good delivery agent for the reporter plasmids. But what about actual applications? And while we are looking into multiple applications of these materials, one of the things that we are trying out, and which is kind of interesting, is that we are trying to see if antifreeze protein encoding plasmids can be delivered topically on the skin. So basically, humans do not have a mechanism of saving ourselves from extreme cold. So if we expose ourselves to extreme cold, we will get what is called frostbite. Whereas there are several other organisms which do express antifreeze proteins. So we are trying to see if we could make these plasmids express antifreeze protein through our skin delivery agent, and we can confer cold hardiness onto the human skin. So this project is going on. Moving on, changing gears a little bit. Well, MGP9 worked very well for us in terms of skin delivery because of its inherent properties. But what about delivery to other organs? So here we chose lungs, which is again an accessible organ. And this work is primarily done by Anupama. And so here, lung again, like skin, very large surface area, although it's internal, but the access is not that difficult. There are multiple ways in which you can access the lungs. And there are a large number of lung diseases which are being, therapeutic applications for these diseases are being looked into using gene delivery applications. But in the lung as well, like in the skin, there are advantages, but there are inherent barriers in the lung as well. And we heard Professor Haynes' talk a little earlier, and he did mention about some of them. And mucus is one of the major barriers that deters nanocomplexes from entering the lung. So these are the epithelial. The green ones are the epithelial cells. So if you have to reach the epithelium, for your gene delivery purposes, you have to cross the mucus layer. And what is this mucus layer? Basically, it's a mesh structure. The mucin polymers form this mesh structure, and they are also glycosylated. So you have both the steric barriers because of the mesh structure, and because of the glycosylation, you have electrostatic barrier as well. And the viscosity in disease conditions is quite high. The normal mucus is also more viscous than water, but in disease condition, the viscosity is high, the pore sizes might be smaller, and so on. So the earlier idea that you could use a mucoadhesive material to release drugs through the mucus into the epithelium is slowly, giving way to generating mucus penetrative systems. So basically, for nucleic acids in particular, where you have to reach up to the epithelium, epithelial cells in order for the nucleic acid to act, you would need mucus penetration rather than mucus adhesion. And I have referred to Professor Haynes' work again here because it's quite seminal in this context. And so what are these mucus penetrating agents? How do you make the nanoparticles or nanocomplexes penetrate through the mucus? So basically, you put different kinds of coatings. So there are inert coatings like PEG, and you have to have a dense coating of PEG. You could also have hydrophilic coating, and basically they take care of the electrostatic barrier. You could use materials like N-acetylcysteine or thiomers, which will cut through the SS bonds and replace the SS bonds in the mucin polymers. You could also use osmolytes like saline or D-mannitol, which will improve the viscosity of the mucus and thereby help in the movement inside the mucus. So, taking all this information together, what we try to do, so we have to overcome both the mucous barrier and the epithelial barrier. So we designed the particle first of all with the peptides, both the CRHC as well as M9 independently, and we use plasmid DNA as well as siRNA. So same principles as I explained earlier to form the core of the particle. So the condensation, release, balance, endosomal escape, and so on. Second thing that we did is we coated it with chondroitin sulfate. Now, chondroitin sulfate is again an anionic polymer, and we have. I don't have time to go into the details of that, but in a series of studies we have seen, and several other groups have established this as well, that if you coat nanocomplexes, DNA-carrying nanocomplexes with a small amount of chondroitin sulfate, the charge comes down and the cellular uptake increases. In some cases, the intracellular routing also changes. And so we thought that, since hydrophilic polymers are known to help in overcoming the mucous barrier, so we used chondroitin sulfate A. We thought that the transfection also will increase because of that. Moreover, it is also amenable to conjugation because of the CO group. And so the third thing that we did is we conjugated CSA with mannitol. Okay, so mannitol will be helping in reducing the viscosity and pushing it through. So with this strategy, we prepared we characterized the complexes in detail. I'm not going into that here. We made the nanoparticles. So we saw that the sizes increase a bit. It goes up to 500 nanometer, but the charge drops. We don't see a lot of change in the siRNA complexes, especially with the CRHC peptide. That could be because the CRHC peptide condensation is slightly poorer than the MGP9 peptide, so that could be one of the reasons. And in case of siRNA, we actually use a slightly smaller amount of coating because siRNA is small, so the charge ratios that we use are different as compared to plasmid. So that could also be one of the reasons why we do not see much of change in that case. But when we looked at the transfection, we do see that we checked it in 2 different cell lines, and we do see that in case of B2B bronchial epithelial cells, there is a large increase in the transfection efficiency, both with the CSA coating as well as with the CSA mannitol coating. Mannitol is actually known to increase caveolo-mediated transport inside the cells. So both CSA as well as CSA mannitol increase the transfection. We don't see much of an effect in case of A549 because it's a cancerous cell. So CS probably, annexin is one of the receptors for CS, and probably the low expression on the cell surface is one of the reasons why it does not show up much. But the mannitol does show high transfection efficiency. So next, we were interested in seeing how does this system behave in presence of the mucous barrier. And for this, we made an artificial mucus that mimics the cystic fibrosis mucus. And we measured the change in viscosity. We measured the ability to diffuse in the mucus. And we also measured how does it penetrate through the mucus. So I'll just show the data quickly on these systems. And first, the diffusion. So we used a nanoparticle tracker analyzer to measure the diffusion of the particles using this equation. And what we see is that in case of the mannitol complexes, both for the plasmid as well as for the siRNA, we see a large increase in case of the M9. We don't see so much of an increase in CRHC2 as we saw in case of the size and charge. But with the mannitol, with M9 peptide, we see a large increase in the diffusion, which is very good for us. When we looked at the viscosity also, we found that, and we measured this at somewhat high shear rate, mimicking the cuff condition. And there also we see that the mannitol coated complexes do show a lot of drop in the viscosity of the mucous viscosity, which again is a positive point for us. And thirdly, when we looked at how the particles penetrate in a transfer system, we we collected the particles from the receptor and over a period of time and saw if they are able to move through the mucus and go into the receptor. And we do see that, yes, in case of the CSM mannitol particles, there is quite a bit of movement, both at 4 hours and also in some cases at 24 hours as well. So clearly the particles are penetrating through the mucus. And a quick check in vivo where we looked at labeled plasmid DNA and intratracheally instilled it in the mouse in the form of nanocomplexes. We see that the CSA mannitol does show stronger signal as compared to the non-coated or the CSA coated particles. So there is much more to do in this sphere. We still don't understand all the properties of the particles in terms of their ex vivo movements in the mucus, etc. But along with that work, we are also trying to see if this would have some application. And one of the interesting things that Anupama thought of was that why not apply this to the disease called silicosis, which is basically an inflammatory disease where an inflammation plus fibrosis, where like if you inhale silica particles for a long period of time or very large amount of silica particles even for a short period of time, you develop inflammation and fibrosis, which is shown by increased collagen deposition and also increased TGF-beta. So what we quickly, in a quick set of experiments, when we deliver a microRNA that is known to act, known to be antifibrotic, we saw that using this delivery system, when we delivered this mRNA, we do see a drop in the TGF-beta levels. We do see a decrease in the collagen deposition. So again, like, this is very preliminary. We need to expand this further, but the mucus penetrating complex does seem to have some effect in terms of lung gene delivery. I think I will stop here, and the students whom I mentioned through the talk, I'm very thankful to them to bring, for bringing the work to this stage. There are several other students whom Anusha, Ujjwal, Sarita, Sabyasachi and all, whose work I have not presented today. And I'm equally thankful to them as well for taking the lab work forward, funding agencies, all my collaborators and the students of the collaborators who actually did the work. And thank you very much for listening in. I'll be happy to take any question if there are questions. Thank you.
Thank you for this and very informative. And now, if participants have any questions, we can take some questions. Okay, so Professor Muniya, I have one question for you.
Yeah.
Like before synthesizing these nanoparticles, how do you because it's a very tiresome process. You synthesize the particle and you go ahead with the mouse model study. So you should have some idea about the efficacy of the drug which you're going to deliver. So what are the parameters you check before going ahead with the mouse model study and synthesizing the nanoparticles? So how do you check the efficacy?
Right. So in case of nucleic acid delivery, which is what we are primarily focused on, we always, first of all, after we make the particles, we make sure that they are monodisperse. They do not aggregate too much. No aggregation is most preferred, and we make those checks thoroughly because, for any activity that you are going to measure, if the particles are not well characterized, then you will not be able to explain the results of your in vitro in vivo assays. And we also check the in vitro transfection efficiency is what we check for nucleic acids, whether they are transfecting the cells in the in vitro scenario. Once we know that, yes, they are the particles are monodisperse and they have all the characteristics that we expect them to have in terms of size, charge, and dispersity. And they also are, in vitro, they are able to deliver the nucleic acids in the cell lines. Only then we move into the animal model. It is not, although I have to say that it is not necessary that something that works in vitro will work in vivo, of course, but also sometimes something that does not work very well in vitro might actually work in vivo. So that's always there, but these are the initial steps that you have to, undertake before you actually move in vivo.
Right, I'm understood. Do we have any other questions from participants? One question.
Yeah, so I think Arpita has asked, can we explore this Na-based
Nucleic acid-based conjugate nanoparticles is or intravascular drug delivery?
So what kind of drug do you want to deliver? Because if it's non-nucleic acid based drugs, then your system has to be somewhat different. So because the principle of using these peptides as a nucleic acid delivery agent is that the peptides have positive charge, the nucleic acid has negative charge. So it's only through electrostatic interaction that they form the nanoparticles. If you have to do a drug delivery, you would probably need to encapsulate the drug, in the particle. So you might have to change the design, choose different kinds of peptides which will self-assemble and encapsulate the drug. So the designs will be somewhat different. Yeah. Any other question?
No, I think the
Okay.
questions we have. Thanks a lot, Haynes.
Thank you. Thank you.
Okay, let's move to the next session.
Ladies and gentlemen, our next session is about where you to kindly mute your microphones and wait. Prepare for the session. Thank you. Ladies and gentlemen, please join me in welcoming Professor Tejal Desai, University of California, San Francisco. Professor Tejal Desai pursued PhD in biomedical engineering jointly from the University of California, San Francisco, and University of California, Berkeley, Bioengineering Department. Dr. Desai is a Deborah Cohen Endowed Professor, Department of Bioengineering and Therapeutic Biosciences, School of Pharmacy and Medicine at UCSF, and Professor in Residence, Department of Bioengineering. He is an accomplished biomedical research specializing in multiple fields, ranging from material science to tissue engineering, and envisioning to seek solutions to existing drug delivery challenges through utilizing concepts of nano and microtechnology. She has won multiple awards between 1994 and 2022, including the American Institute of Medical and Biological Engineering, Fellow of the Biomedical Engineering Society, Fellow of the Controlled Release Society. Please join me in welcoming Professor Tejal Desai.
Hi, good afternoon. My name is Tejal Desai, and I'm professor and chair of the Department of Bioengineering and Therapeutic Sciences at UCSF. And today I want to talk to you about some of our work in developing cardiovascular devices using micro and nanostructured materials to modulate the tissue microenvironment. To begin, I wanted to give you an overview of the Desai lab, where we're interested in developing advanced biomaterials for therapeutic applications. And in this regard, we are interested in how we can manipulate the topography, morphology, and chemistry of materials in order to enhance therapeutic delivery across a range of physiological systems, including tissue barriers, cell-based trafficking, and the immune system. And over the last many years, we have developed a number of different materials at the micro and nanoscale, where we've really tried to understand how those different length scales can interface with biology. We work in everything from creating micro devices for oral protein delivery to microscale rod structures in order to mitigate fibrosis. But today I'm going to focus on some of the work that we're doing in developing devices that interface with the cardiovascular system. So we're interested in this concept of structure. How can you take a bulk material and change the interface so that it has a more intimate and better contact with the microenvironment? And so we've developed some technologies involving both peravascular wraps as well as stent, where we're trying to make sure that the materials don't just stay inert, but really interact with the environment in order to resolve inflammation and promote healing. So most of you are familiar with the concept of a bare metal stent. These are used routinely in the area of cardiovascular medicine, whereby you want to open up a blocked or clogged artery. And these metal stents are generally made of materials such as cobalt chromium, stainless steel, or nitinol, and sometimes coated with a biopolymer ceramic. Several years ago, we moved from bare metal stents to what we call drug-eluting stents. And we did this because it was shown that over time, when you put a bare metal stent into the body, the vessel undergoes restenosis, or an eventual clogging of the vessel again. And that was facilitated by an overgrowth of smooth muscle cells and deposition of materials that would start to close up the vessel. And so by incorporating drug elution onto the stent, in particular anti-neoplastics or anti-proliferative agents, one could have a stent delivered in the vessel that would stop this proliferation of smooth muscle cells. This seemed like a good idea, but what we discovered is those same agents that stop the proliferation of smooth muscle cells also do damage to the endothelial cells in the area, causing an inability of the vessel to completely heal. And so what is restenosis? Well, this is a phenomenon where the vessel or the lumen of the vessel actually gradually starts to narrow. It's characterized by granulation tissue, including infiltrate of macrophages, the successive proliferation of smooth muscle cells, the deposition of a proteoglycan matrix. This eventually leads to this closing or narrowing of the vessel and thrombus formation. And you can see in the upper right-hand corner the image of a stent that's being encapsulated or surrounded by this overgrowth of tissue, which then leads to complications later on whereby the stent has to be removed and the actual problem that we were trying to fix becomes another problem. So we were interested in, could we create the ideal stent surface? Something that was non-inflammatory, that promoted endothelial cell growth, but also inhibited smooth muscle cell growth. And so drug-eluting stents, as I mentioned, are able to inhibit smooth muscle cell growth, but actually they don't promote endothelial cell growth. And so we wanted to see, could we actually just tune the architecture of the stent, the surface that makes intimate contact with these cells, in order to promote endothelial cell growth and inhibit smooth muscle cell growth without the use of pharmacologic agents. And so we did that. We actually developed processes whereby you could use a two-step anodization method in order to grow what we call titanium nanotubes on the surface of three-dimensional objects, including stents. And you can see in this electron micrograph that we can get these fairly uniform coatings across the material. And these again are at the nanoscale. So from a macro perspective, all you see is sort of this roughness. But if you zoom into it, you can see these discrete tubes that are patterned all over the surface. And you might ask, why nanotubes and why not something else? Well, in a number of studies, we showed that nanotubes actually have profound biological impacts, in particular with smooth muscle cells and endothelial cells. And so what we showed is that proliferation increases in endothelial cells that are in contact with nanotubes, but actually decreases in the smooth muscle cells that make contact. And you can see this in these two graphs looking at endothelial cells and vascular smooth muscle cells, that cells actually are fairly constant between flat and nanotube surfaces in the case of endothelial cells, but dramatically reduced in the case of smooth muscle cells. We went on to show that this is a phenomenon that really changes the expression of genes that promote specific processes. And in particular, for smooth muscle cells, they change genes or downregulate genes that dictate proliferation and migration. They also downregulate the inflammatory genes that are associated with these processes. In contrast, if we look at the endothelial cell nanotubes, we see genes that are upregulated for things like proliferation and migration, still maintaining a downregulation of inflammatory genes. Now, this is exactly what we wanted to see in terms of developing a material that could interface long-term with the vascular barrier. We went on to show that there are several key transcription factors that are altered by the presence of nanotubes. These are implicated in things like migration, integrin activation, and mechanotransduction. And looking at the two cell types side by side, we see that vascular endothelial cells have this stark increase in proliferation and migration, whereas smooth muscle cells absolutely don't. This really demonstrated to us pro-healing properties that these types of structures could have on arterial cells. So, of course, it's nice to show things in vitro and differential effects that we observed in our cells, but the real test is what happens in vivo. And so over the last several years, we've been looking at trying to produce studies that can give us a proof of concept about, can we take stents and pattern them to induce a more pro-healing response? And so our first preclinical study was that of a in vivo rabbit model in the peripheral artery. And in this model, we actually looked at 3 different types of stents: a stainless steel stent, an uncoated titanium stent, and then a titanium nanotube coated stent. And in this regard, we were able to look at both the effect of material changes as well as the nanostructure. And these stents were deployed into an injury model that was produced through overinflation of the artery and studied for about 28 days. And what we saw was quite striking, in that the titanium nanotube surfaces, the stents that had that nanotopography, actually had the most reduction in stenosis in these rabbit models. And you can see here, these are the 3 different types of stents that we implanted: stainless steel, titanium, and nanostructured titanium. And if you look at the bottom panel, you can see this restenotic process that's undergoing in the artery, whereby you get that excessive tissue that's encapsulating the struts of the stent. And so by looking at the thickness of that layer, we can judge the extent by which restenosis is happening. And yes, indeed, you can see that compared to stainless steel, our titanium nanostructures are significantly lower in terms of the restenotic lesions. We also repeated these studies in a preclinical porcine coronary model. And here we really wanted to go against and sort of look at one of the stents that is used clinically. And in this case, this was a stainless steel Boston Scientific stent, where we wanted to look at our titanium nanotube stents compared to those. And so again, we deployed these in this case into the left circumflex or left anterior descending coronary artery, looked at this over 28 days, and wanted to see what type or the extent of restenosis that was happening. And again, we saw this stark difference between the stainless steel and these nanotube coated stents, where the ones that had nanotube not only had decreased stenosis and decreased neointimal growth, but actually more important, or just as important, actually had full endothelialization across the surface, which was really important because, as I mentioned at the beginning, we want to promote endothelial cells and we want to stop smooth muscle cell growth. And so we're really excited about taking this concept and trying to move it forward, looking at ways in which we can take materials that otherwise might cause injury or inflammation and subtly changing their outer structure in order to promote a healing response. And here's just a short video I want to play showing this concept. This is what the stents look like. These are the ones that we designed in-house. And you can see the whole entirety of the stent is actually covered with these nanotubes. And so the thought is that if the nanotubes can promote migration and endothelialization, you get this coverage by endothelial cells that can then sort of coat the surface with a cell layer. And this cell layer then stops the smooth muscle cell layer from actually overgrowing. And what we saw when we explanted these stents was that you can see this intimate contact between the cells and the titanium nanotube material itself. And if we zoom in even further, you can see that the cells are actually grabbing on to these tube-like structures and interdigitating to some extent. And what we also found is that they were secreting things like fibrin, which are known to be important for resolution of vascular injury. So we're excited about these dynamic interactions that we see between the tubes and the cells, and in particular, how we can regulate cell phenotype in response to synthetic materials. Now, the second project I want to talk to you a little bit about is also in the realm of cardiovascular intervention. And one of the things we know is that inflammation is the Achilles heel of any cardiovascular intervention, in the sense that it's a response to vascular injury. And when we put in devices such as angioplasties or stents or do surgical bypass, we're essentially creating a vascular injury that leads to inflammation. So regulating this process remains a major unmet scientific need within the vascular surgery realm. So in the last couple of years, working with Michael Conti, who is Chief of Vascular Surgery at UCSF, in thinking about the application of pro-resolving therapeutics. Now, traditional anti-inflammatories are very effective in that they lower the magnitude of inflammatory signaling. And so we can go from an inflammatory state where we have onset and then resolution over to something in where we have a much lower magnitude of inflammation, although the time duration is about the same. Pro-resolving therapeutics, on the other hand, actually drive the immune system towards a more rapid resolution of inflammation, shifting the curve to the left, where we have inflammation, but it gets resolved much more quickly. And so we were very interested if we could actually take these pro-resolving molecules and apply them in a well-controlled way to cardiovascular injury. There's a whole range of pro-resolving therapeutics, ranging from RVD1 to RVD5. And all of these have been implicated in different disease models, ranging from lung injury to colitis to neuroinflammation. But in particular, we were interested in something called Resolvin D1, or RVD1. And this is because it's been shown to be a very effective vascular therapeutic. RVD1 is naturally synthesized from enzymatic action on DHA. And Michael Conti's group, as well as some others, have shown that if you can apply this as a gel or continually drip it onto the vessel, it can attenuate inflammation in a traumatized vasculature. So this leads to possible applications in everything from AV fistulas and grafts to ischemic injuries. And you can see from the images on the right, a vessel that's untreated, and you can see this intimal hyperplasia that's forming in the middle, versus one that's treated where you really resolve that excess inflammation and tissue generation. So we were very interested in creating an RVD1 eluding biodegradable film, where we could locally deliver RVD1 to sites of surgical or vascular injury. But we had to think about what the design criteria was for doing so. And the key was to develop something that was really thin, that could go around a vessel from the outside and be able to stay there, deliver the drug, but deliver it in one direction such that most of the drug is targeted to the site of injury. In order for translation, we wanted this to be a polymer that had an established safety profile, and most importantly, could keep our active compounds stable. So we've developed what we call a biodegradable thin film wrap. And this wrap was designed to have multiple layers, such that there was an impermeable layer, and then a layer which allows drug to actually penetrate out. And in doing so, we could wrap this film around an artery and have RVD1 come out unidirectionally towards the walls of the artery. And the graph on the bottom right shows exactly that, where upon elution, we see elution happening only towards the inside, but very little coming out of the outside. And these films are very thin, less than 30 microns, and almost act like a saran wrap, which I'll show you. And what we showed in a perivascular model in an acute arterial injury is that these types of wraps can be very effective. And so on the top three panels, A, B, and C, I'm showing you an angioplasty where you see this resultant injury that occurs in the vessel. Then a vehicle where we're just putting a wrap That actually does improve a little bit of the healing response in it of itself. And then our RBD1 wrap on the upper right-hand corner, where we see this resolution of inflammation and a decrease in neointimal area that's been formed. And this does hold true when we look at the actual data and measure the amount of neointimal coverage. Indeed, RBD1 wraps are able to decrease compared to vehicle and angioplasty alone in a significant way. We also looked at this in a rabbit model and looked at hyperplasia at 28 days. And what we saw is that, again, local RBD1 treatment reduces vein graft hyperplasia in a significant way. And so in this case, we actually decided to make our wrap even thinner, so about 10 microns. And you can see it has two layers, an 85% PLGA layer and a 50% PLGA layer. The 85% layer, again, is impermeable to drug release. The 50% layer actually allows the RBD1 to come out towards the site of the vessel. And so we show that with the RBD1 wraps, we can get a significant healing response where neointimal thickness is diminished and overall vessel thickness as well. What was really interesting is that in that local area, we found that leukocyte infiltration as well as macrophage infiltration was also reduced dramatically, which makes sense because those are the hallmark cells of inflammation. And we know that RBD1 actually works in concert to facilitate a resolution. So we've been working hard over the last year and a half or so in trying to scale up this technology. It's one thing to create a thin film wrap in the lab, but it's another thing to actually manufacture, scale, and GMP process it. And so with the help of a senior scientist in my lab, Dan Bernards, we've actually created these GMP compliant wraps that are packaged. They actually incorporate the RBD1 within them, and they're foiled or packaged in order to provide an oxygen and moisture barrier, and also labeled so that we know which side is up and which side is down, because again, they are there to provide asymmetric delivery. And so I'll just a little bit about what these look like. Again, very, very thin, like saran wrap. And these go right around the vessel. And this is Dr. Conti, who's placing these into a model. This is our actually an ovine model or a sheet model where we're trying to scale these wraps up to. And you can see it just sort of tucks right in. We don't need any sutures. We don't need any adhesives. Because of its thin nature, it actually is normal. And so we're excited to see how we can bring this forward and hopefully begin the process of translating it out of the lab and into a product. So I know I'm out of time, but I just want to end here by highlighting the fact that we can use our ability to tune micro and nanoscale structure of materials to modulate the body's response and hopefully improve healing to materials or devices that are placed within the body. I didn't have a chance to talk to you about a lot of the other work that we're doing in the lab involving drug delivery, immune modulation, and fibrosis. But again, the key concept is taking a material and really being able to manipulate its structure at the micro and nano scale. And with that, I want to end. I appreciate your time in watching this, and I want to I acknowledge my lab at UCSF. That's been a wonderful team to work with and really move a lot of our ideas forward. And of course, I want to thank our funding sources, in particular, NIH and the VITA program for the work on the perivascular wraps, the Al Mann Institute for facilitating our work on the stents, and all of our other funders for helping us think about new innovations at the interface between engineering and medicine. Thank you